Okay, so welcome to this next video in which we are discussing Phosphoinositide 3-Kinase enzymes. Now, we're currently focusing our attention completely on type 1a phosphoinositide 3-Kinase enzymes, which remember consist of a catalytic subunit, which is either P110 alpha, P110 beta, or P110 delta, along with a regulatory subunit, which is either, it's one of these five up here. It's P50 alpha, P55 alpha, P85 alpha, or P85 beta, or P55 gamma, so it's one of those five. Okay, right, and all of them pretty much have this same structure here. There's a question of whether you have these two additional domains here, but these don't interact with the catalytic subunit, so it doesn't really matter, they're just sort of dangling off the side anyway. Okay, right, so we now want to see how these type 1a PI3 kinase enzymes are going to be activated by receptor tyrosine kinases. Okay, so we're now going to turn our attention to receptor tyrosine kinases and how they are activated and how they are then going to activate the type 1a PI3 kinase enzymes. Okay, so receptor tyrosine kinases then, they are a large family of receptors. Okay, there's around 58 known receptor tyrosine kinases in humans alone. Okay, and for short, receptor tyrosine kinases are often abbreviated to RTKs. R for receptor, T for tyrosine, and then K for kinase. Okay, and the reason that they're called that is because they have an inbuilt tyrosine kinase. Okay, they are tyrosine kinase enzymes. Okay, so let me draw you a picture of the basic structure of most receptor tyrosine kinases. Now, not all of the 58 receptor tyrosine kinases in humans conform to this structure that I'm about to draw, but most do. There are a few exceptions, such as the insulin receptor, but apart from a few tiny ones that don't conform to this, most of the other ones do. Okay, so they are a single polypeptide, so here is the amino terminus of that polypeptide. Then the first important domain that you have, which is on the extracellular side of the cell membrane. And by the way, when I draw these two parallel lines here now, which I'm now even colouring in for you in green, these now represent the inner and outer leaflet of the cell membrane, rather than, for instance, a double-stranded piece of DNA anymore. Okay, so this extracellular domain that we've got here is what's known as the LBD, and the LBD stands for the ligand binding domain. Okay, so this is where the receptor tyrosine kinase is going to bind to the ligand molecule, which will be on the extracellular side of the cell membrane. Okay, right. After the ligand binding domain, then, you have a membrane spanning alpha helice. Okay, or an alpha helix rather, since we're not talking about the plural. Okay, so that's an alpha helix that is spanning the membrane there. Okay, and it will contain a large number of hydrophobic residues which will interact nicely with the hydrophobic core of the cell membrane here. Okay, then on the intracellular side, what you have is the domain which makes up the tyrosine kinase. Okay, so this is the tyrosine kinase domain, and then after that you have the carboxylic acid tail. Okay, right. So let's colour in these different domains. So we'll start with this domain here. So this is the tyrosine kinase domain. Now, in some receptor tyrosine kinases, the tyrosine kinase domain is what's known as a split tyrosine kinase domain. Okay, so let me just explain what this means. So, if you look at the linear structure of this polypeptide, if you look at the primary structure of it, in some receptor tyrosine kinases, there is one cohesive portion that will then fold up into the tyrosine kinase domain. Okay, so there's one section of the primary structure of the polypeptide which will then fold up to produce the tyrosine kinase domain in the tertiary structure. Okay, so that's a whole tyrosine kinase, okay, an unsplit tyrosine kinase. Okay, Whereas in some other receptor tyrosine kinases, you have what's known as a split tyrosine kinase, which is where if you look at the primary structure, there are the portions which are going to fold up to make the tyrosine kinase domain are separate, basically. Okay, so you have this portion in the primary structure here in blue, and then you have a portion in between that which isn't going to contribute to the tyrosine kinase domain, and then a portion after that which is. So these two portions in the three-dimensional structure, so once it's folded up, these will come together to make the tyrosine kinase domain. Okay, so this now is known as a split tyrosine kinase domain. Okay, and the portion that sits in between the two halves in the primary structure that are going to make up the tyrosine kinase domain in the tertiary structure, this is known as the kinase insert. So this little section in the middle here, this is called the kinase insert. Okay, because it's inserted in between the two portions which will fold up to make the tyrosine kinase domain. Okay, we'll also then colour in the ligand binding domain in orange here. Okay, so these are the main two important domains of a tyrosine kinase receptor, or a receptor tyrosine kinase rather. You have the ligand binding domain extracellularly, which is where you bind your ligand, and then you have your tyrosine kinase domain intracellularly. Now, I said that there were 58 different receptor tyrosine kinases within humans. Okay, now these are classified into families. Now we are not going to go through all the families because there's actually 20 of the things. Okay, I'm going to introduce you to one of the families so that we have something, so that we have a nice specific example, okay, to be working with. So the family of receptor tyrosine kinases which I'm going to introduce you to is what's known as the ERB-B family of receptor tyrosine kinases. Okay, now the ERB-B family of receptor tyrosine kinases contains 4 different receptor tyrosine kinases. Okay, these 4 different receptor tyrosine kinases are called ERB-B1, ERB-B2, ERB-B3, and ERB-B4. So it's got a nice simple naming system. Okay, right. There are other names, unfortunately, for all of these proteins as well. Each of them has another name, which is that they're also called HER receptors. Okay, so luckily the numbering is the same. So ERB-B1 is also called HER1, ERB-B2 is also called HER2, ERB-B3 is also called HER3, and ERB-B4 is also called HER4. Okay, now what does HER stand for? Well, HER stands for Human Epidermal Growth Factor Receptor. Okay, and I'll abbreviate epidermal growth factor to EGF for now. Okay, so the H is for human, the E is for epidermal growth factor. Okay, so here's the H for human, the E is for epidermal growth factor, so this tells us beautifully what the ligand for all of these B receptors is, and then the R is for receptor, so HER receptors. Okay, now, the ERB-B1 or HER1 receptor also has another name. It's also known as the epidermal growth factor receptor, so it is the epidermal growth factor receptor, so it's the one with the highest ranking basically, but HER2 is also very important. It's the one that's upregulated in some forms of breast cancer. Okay, so this stands for, I think I should write this out in full at least once, the epidermal growth factor. Okay, so that's the ligand that we're working with, which from now on will be abbreviated just to EGF, and then the epidermal growth factor receptor. Okay, right, so let's colour this in red here. Okay, so let's have a look at the structure then of these ERB-B family receptor tyrosine kinases. Okay, so I've discussed the basic structure of all receptor tyrosine kinases, but the ERB-B family has specific characteristic features that are special for it, okay? So I'd like to just give those special features. Now, What varies between the different families of receptor tyrosine kinases is generally the ligand binding domain. So the ligand binding domain will have very different structures between the different families of receptor tyrosine kinases. And this is because generally the receptor tyrosine kinases in different families have different ligands. Okay, so the ligand binding domain needs to bind a different ligand, and therefore clearly it needs to have a different structure. So all of the B family receptor tyrosine kinases, these 4 members here, they all have a certain structured ligand binding domain, and I just want to discuss this now. So once again, here is the amino terminus of the polypeptide. Then, this time, the ligand binding domain is going to have 4 important domains. Okay, so this rectangle still represents the ligand binding domain, but I'm now going to separate it into 4 separate domains. Okay, then it's going to span the membrane. So here is the phospholipid bilayer. It's going to span the membrane, and then it's going to have the tyrosine kinase domain intracellularly here, and then we'll have the carboxylic acid tail intracellularly here as well. Okay, so once again, let's colour in the domains that we know. So here is the tyrosine kinase domain. Okay, I think I'll start colouring in the membrane-spanning alpha helix in red. I've stopped drawing it actually in the alpha helical structure because that's a pain to have to draw. So just remember that that is an alpha helical structure that's spanning the membrane there. Okay, and then the ligand binding domain has these 4 separate domains. Okay, so 2 of these I'm going to draw in turquoise, or colour in turquoise rather. Okay, and these 2 domains in turquoise, these are going to be the leucine-rich domains. Okay, so these portions are rich in leucine. Okay, now what is the amino acid leucine? Well, let me just remind you of what the amino acid leucine is. Okay, so this is a leucine-rich domain. Okay, so I will draw this as a residue rather than as the free amino acid. Now what that means is I'm going to draw the amino acid as though it's bound to the amino acid in front of it and also the amino acid after it. So here is the amino group, okay, of the amino acid, and this bond represents the bond to the carboxylic acid group of the amino acid in front of it. Then here's the alpha carbon with a hydrogen coming off it, and then here is the carboxylic acid group, which will then be linked to the amino group of the next amino acid along. Okay, and the R group then of leucine consists of a methylene group, like so, then with another carbon coming off this, which then has 2 methyl groups coming off it, one like so, another here, and then a hydrogen coming off it. Okay, so this is the structure of the amino acid leucine, drawn as an amino acid residue. Okay, so drawn as though it's within a polypeptide rather than just as a free amino acid. Okay, so these leucine-rich regions that we've got here, these are going to be rich in leucine. So you're going to have a large proportion of leucines in there, a larger proportion than would just occur by average. Okay, by random chance. Okay, then these 2 other domains that I've now coloured in purple here, these are cysteine-rich regions. Okay, so they're going to have a large proportion of cysteine residues within them. Okay, so you find this in all of the 4 B receptors. They all have this alternating leucine-rich region and then a cysteine-rich region. Okay, so let me just remind you of the amino acid structure for cysteine. Okay, so once again, draw the core structure of the amino acid. Here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, okay, and then the R group of cysteine consists of a methylene group with a phyl group coming off it. Okay, so this group here where you have a sulfur atom bound to a hydrogen atom, that's called a phyl group, and it has similar chemical properties to alcohol groups, okay, because sulfur is in the same group of the periodic table as oxygen. Now clearly if that sulfur was an oxygen, that would be an alcohol group. Now since sulfur is in the same group of the periodic table as oxygen, it has similar chemical properties to oxygen, and therefore the phyl group is going to have similar chemical properties to the alcohol group. Okay, so this is cysteine. Right, so in the ligand binding domain then of all B family receptor tyrosine kinases, you have these alternating leucine-rich domains and cysteine-rich domains. Okay, and that goes for all of the B family receptor tyrosine kinases. Okay, now we've got a nice example of receptor tyrosine kinases. We're going to discuss the activation of receptor tyrosine kinases, okay, focusing specifically on, let's say, the epidermal growth factor receptor, ERB1, or HER1. Okay, right. Now I'm going to demote it. Don't want to have to continually draw these 4 different domains. I'm just going to go back to representing this as one orange blob called the ligand binding domain. But be aware that it is really alternating leucine-rich and then cysteine-rich, leucine-rich, etc. Okay, right. So the first thing then that happens is that epidermal growth factor is going to now come along and bind to the ligand binding domain of the receptor tyrosine kinase. Okay, so here's the amino group, here is the membrane spanning alpha helix, this is the ligand binding domain, and then we've got our our tyrosine kinase domain intracellularly here. Oh, and something else I should have mentioned when discussing the ERB receptors is that all of them have whole tyrosine kinase domains. Okay, so the portion which is going to fold into the tyrosine kinase domain is all in one section of the primary structure rather than being in 2 split sections like so with an insert in between them. Okay, so it's a whole tyrosine kinase domain, always in the ERB family receptor tyrosine kinases. Okay, right. So once again, here is our tyrosine kinase domain, and we haven't yet discussed what tyrosine kinase enzymes actually do, so I will discuss that in a moment. Okay, then we've got our ligand binding domain here in orange. Okay, and then we've got our ligand now bound to the ligand binding domain here in turquoise. Okay, and of course our ligand in the case of the epidermal growth factor receptor is going to be epidermal growth factor, EGF. Okay, so what's now going to happen is that this epidermal growth factor receptor, which has got epidermal growth factor bound to it, is going to dimerize with another epidermal growth factor receptor, which has got epidermal growth factor bound to it. Okay, now there is a little bit of room for forming heterodimers. Okay, it's not necessary for the epidermal growth factor to only be able to bind to another epidermal growth factor receptor. Okay, it could bind to one of the other B family members. Okay, so let me explore that a little bit more after I have drawn a picture of it. Okay, right, so here is the amino group of this second receptor tyrosine kinase here. Here is the tyrosine kinase domain down here. Okay, and then here is the C-terminal tail here. Okay, so let's colour in the special domain. So here is the tyrosine kinase domain. Okay, here is the ligand binding domain, and of course this other receptor tyrosine kinase that we have dimerized with is going to have ligand bound to it as well. So now the question comes, which receptor tyrosine kinase can we dimerize with? Okay, so let's say that this first original one here is an epidermal growth factor receptor, okay, or an ERB1 receptor. Which receptor tyrosine kinases can it dimerize with? Well, first thing to say is that it can dimerize with an absolutely identical one to itself. Okay, so it can bind to another epidermal growth factor receptor or an ERB1 receptor, which also has epidermal growth factor bound to it. That is known as a homodimer. Okay, homo means same, dimer means 2-membered structure. Okay, so this is a 2-membered structure where the 2 members are identical, they're the same. Okay, alternatively, this second receptor tyrosine kinase can be a different one. Okay, but it must be within the same family so that they have similar ligand binding domains to one another. Okay, so it can be one of these other B family receptor tyrosine kinases, but it cannot be just any old receptor tyrosine kinase. Okay, it can't be, for instance, a vascular endothelial growth factor receptor. It has to be one of these other B family members. Okay, and when it dimerizes with another family member which isn't identical to itself, that's called a heterodimer. Hetero means different, dimer once again means two-membered structure. So the two members in the two-membered structure are different in the case of a heterodimer. Okay, so we'll imagine that it's a homodimer, that both of them are identical epidermal growth factor receptors with epidermal growth factor bound to them, because it's simpler. Okay, right. Now we have to discuss what a tyrosine kinase actually is. So I think we'll start off with that. What are these domains actually capable of catalyzing? Okay, well, they're capable of catalyzing the phosphorylation of tyrosine residues within proteins. So let's draw a tyrosine residue. So once again, here's the amino group, here's the alpha carbon, here's the carboxylic acid group, and this is the core amino acid structure. The R group then of a tyrosine residue consists of a methylene group, then with a benzene ring coming off it, like so, and then an alcohol group coming off the fourth carbon of the benzene ring. Okay, so this is a tyrosine residue. Okay, right. Now, what then are tyrosine kinase enzymes going to catalyze? Well, they're going to catalyze the phosphorylation of this alcohol group coming off the benzene ring of the tyrosine residue. Okay, and I think I'm going to have to pause this video here and we'll continue on after I've had a glass of water.